Look at this vial of insulin. We rely on medications like this to save lives every single day. But we don't mix these chemicals from scratch. We manufacture them by hacking a biological instruction manual. This model represents the physical structure that holds that instruction manual, the famous DNA double helix. To read the code clearly, we unwind that three-dimensional shape into a flat ladder. Its rungs are 4 chemical bases locking in strict pairs. Apples fall in the tree, binding A to T, and cars park in the garage, locking C to G. Every advancement in genetic engineering begins with these 4 pairings. If you can predict exactly how these bases connect, you can begin to map out and modify the entire human sequence. Having the manual is step 1. Actually reading it requires cellular machinery. When a cell scans a sequence of bases on the DNA ladder, it processes them strictly in groups of 3. Every specific triplet acts as a direct call for a corresponding building block, known as an amino acid. Once summoned, those individual blocks link into a continuous chain, break away, and fold themselves up into a complex 3-dimensional structure. That folded shape is a functional protein, like human insulin. The linear sequence of abstract letters directly dictates exactly how that physical medicine is built. On your biology exam, you must explain exactly how we transfer that specific insulin recipe from a human cell into a bacterial host. We start by targeting the human gene for insulin and extracting it using chemical tools. Next, we extract a circular loop of DNA, called a plasmid, from our target bacterium, and we slice it open with those exact same tools. We have an open bacterial vehicle and a human gene snippet. Say the immediate next step out loud. We place the human code directly into the gap of the bacterial plasmid and seal the edges together using gluing enzymes called ligase. That newly fused loop is defined as recombinant DNA. Using that precise vocabulary is how you secure maximum marks on your paper. Now, we take that completed recombinant plasmid and insert it right back into the empty bacterial host. Placed inside an industrial fermenter, that single bacterium divides rapidly. Because it holds the hacked instructions, every new cell actively manufactures and pumps out usable human insulin. Looking at this photograph of technicians working at a laboratory bench, you are seeing the environment where this biology is applied. This specific extraction and insertion sequence is exactly how working scientists engineer living organisms to produce global medicine. This chart is the consolidated summary you need to answer genetic engineering exam questions. It maps out the base pairings, the protein translation, and the exact steps to cut, open, glue, and insert. To get the full set of revision charts for your May 2026 exams, send me a direct message with the word blueprint. Navigating this topic isn't about memorizing random facts. It's about understanding the specific mechanics of the process. When you can trace the code from a human cell into a functioning bacterial factory, you have mastered the logic the examiners are looking for.